dopamine receptors are mainly of 5 types, D1 to D5. The D1 receptors are present in the striatum, nucleus accumbens, and the frontal cortex, whereas the D2 are more evenly distributed. The D3 fibers are mainly in the limbic area, and D4 in the frontal cortex, amygdala, hippocampus, and hypothalamus. Now, the typical antipsychotic drugs either block both the D1 and the D2 receptors, like chlorpromazine, or block only the D2 receptors, like haloperidol. But this blockade is not specific to any particular area. So they can produce, by blocking the D2 receptors in one area, they can produce a lot of side effects which are unwanted. The atypical antipsychotic receptors block only the D2 receptors in specific areas. The clozapine, which is an atypical antipsychotic, specifically blocks the D4 receptor. Now, dopamine has been implicated in a number of the pathophysiology of a number of neuropsychiatric disorders. We know that psychostimulant drugs enhance the dopaminergic transmission in the mesolimbic system, like I already told you, cocaine and amphetamine. And hence, dopamine has been implicated in the development of addiction. In schizophrenia, of course, the dopamine hypothesis was the oldest one. So now other neurotransmitters are being increasingly implicated. In the classical hypothesis, schizophrenia occurred due to reduced cortical dopamine transmission, that is hypofrontality, with overactivity in the subcortical dopamine. The dopamine has also been implicated in the number of other neurodegenerative disorders and in Tourette syndrome and ADHD. Mainly this role has been derived from because the drugs which are used in the treatment of these disorders act mainly on the dopamine receptors. For example, haloperidol in Tourette syndrome and methylphenidate in ADHD. Like I said, that amphetamines block the reuptake of dopamine. And high doses can be addicting, but low doses are used for the treatment of ADHD. Now coming to the norepinephrine pathways, basically the nuclei of the norepinephrine pathway are two. The cell bodies are located in the locus coeruleus and the tegmental area of the midbrain. From here, they innervate almost all areas of the brain. The locus coeruleus sends fibers to the forebrain, thalamus, and the cerebellum, whereas the lateral tegmental area sends fibers mainly to the hypothalamus and the basal forebrain. You can see in this diagram that almost all areas of the brain are innervated, from neurons originating in these two areas. Now, the locus coeruleus neurons have two activity modes. The tonic firing mode, which is basically responsible for the sleep-wake cycle, and the burst firing mode, which occurs in response to an arousing stimulus, and which is responsible for the attention and vigilance when there is a sudden rousing stimulus. The locus coeruleus neurons play an important role in all the prefrontal cortical functions, including vigilance, attention, working memory, and the executive function. Coming to the adrenergic receptors, just a brief overview. There are three main types of adrenergic receptors: the alpha 1, the alpha 2, and the beta. The alpha 1 and beta are postsynaptic, whereas the alpha 2 are both presynaptic and postsynaptic. So they also function as autoreceptors. Alpha 1 has three subtypes: alpha 1A, 1B, and 1D. Alpha 2 has four subtypes: 2A, 2B, 2C, 2D, and beta has two subtypes: beta 1 and beta 2. Now, the importance of these receptors in treatment mainly accounts for their agonism and antagonism by certain drugs. For example, phenylephrine and xylomethazine are mainly agonists of the alpha 1 receptors, and alpha 1 receptors are mainly responsible for the dilatation and constriction of blood vessels. So, agonists which can constrict the blood vessels include phenylephrine and xylomethazine, which are used as nasal decongestant or as pressure agent. Antagonist is prazosin, which is used in the treatment of hypertension, and because alpha 1 receptors are also located in the CNS, it has been used in the treatment of PTSD. Alpha 2 receptor agonists include clonidine, which is used for opioid withdrawal and ADHD, and guanfacine, which is selective for alpha 2A, which has been used for the treatment of Tourette's and in treatment of ADHD. Yohimbine, which is an antagonist of alpha 2 presynaptic alpha 2 receptors. Remember, these alpha 2 receptors are autoreceptors. So, yohimbine, which blocks the alpha 2 receptors, yohimbine, which blocks the alpha 2 receptors, can induce panic. The beta receptors, the agonist is salbutamine, which is used in the treatment of asthma, and antagonist prototype is propanol, which is used for treatment of social anxiety, because it reduces the sympathetic overactive. Coming to the serotonergic pathways, the neurons for the serotonergic pathways are located in the rapid nuclei of the brain, the midline rapid nuclei. Three main groups: the dorsal, medial, and the caudal rapid nuclei. Their innervation again is widespread to many areas of the brain, including the cortex, the thalamus, the hypothalamus, the cerebellum, down into the medulla and the spinal cord. Serotonin is synthesized from tryptophan. It plays an important role in the regulation of mood, anxiety, cognition, feeding, the sleep-wake cycle, and sexual behavior. And because of this, because it plays a role in so many emotions and behaviors, it has been implicated in the pathophysiology of a large number of psychiatric disorders, including schizophrenia, mood disorders, and anxiety. Now, the tricyclic antidepressants, the SSRIs, SNRIs, they inhibit the reuptake of serotonin into the presynaptic terminals. Of course, PCAs are non-specific. They even inhibit the reuptake of dopamine and adrenaline. And the mechanism, in addition to reuptake of dopamine, the serotonin reuptake mechanism has been, inhibition of reuptake has been proposed for the antidepressant effect. Amphetamines stimulate the release of monoamines, including serotonin, into the synapse. Coming to the serotonin receptors, there are seven types of IHT receptors: IHT1 to 5HT7. Each has subtypes. The 5-HT1 has 5 subtypes, A to E. Buspirone is a partial agonist of the 5-HT1A receptor. It's used as an anti-anxiety agent, and this action at the 5-HT1A receptor accounts for anti-anxiety properties. 5-HT1B and 1D are mainly autoreceptors, that is, they are present on the presynaptic cell. Sumatriptan is a agonist of the 5-HT1B, B, and F receptors. It's used in the treatment of migraine. So, these receptors have been implicated in the pathophysiology of migraine. 5-HT1E is rich in the hippocampus, and because the hippocampus plays an important role in memory formation, this receptor is a possible drug target, new drug discovery target for Alzheimer's disease. 5-HT2 has 3 subtypes, A to C. Buspirone, I have already told you, is a partial agonist of 5-HT1. Now, 5-HT2A is widely distributed, especially in the cortex. Drugs like LSDs and psilocybin are 5-HT2A agonists. And probably this agonism accounts for some of their hallucinatory and psychotic inducing properties. The second generation antipsychotics block the 5-HT2A receptors. So that is why they are believed to be effective in the reducing the symptoms of schizophrenia. Downregulation of 5-HT2A receptors is one of the important mechanisms proposed by which SSRIs exert their antidepressant effect. So 5-HT2 receptor has a lot of clinical significance. In the 5-HT2C receptors have a widespread distribution, and the weight gain associated with olanzapine and clozapine is due to the blockade of 5-HT2C receptors. The 5-HT2C receptor also regulates the dopamine release in the midbrain, including the nucleus accumbens. And because of this, it is a target for developing new drugs for treatment of substance abuse and schizophrenia. Five HT3 receptor, in exception to all the others, is actually an inotropic receptor, and ondansetron, which is an antiemetic agent used in cancer therapy, is a 5-HT3 antagonist. 5-HT6 is present mainly in the limbic and cortical region, and italopridine, which is a 5-HT6 antagonist, has been shown to improve the cognitive performance in Alzheimer's. 5-HT7 has a role in cognitive processes, and also several antipsychotic and antidepressant drugs have an affinity for the 5-HT7 receptor. Lurasidone particularly has a high affinity for the 5-HT7 receptors and is used both in the treatment of mood disorders and schizophrenia. Coming to the cholinergic pathways and acetylcholine, the cholinergic pathways basically arise from two main neuronal bodies: the basal forebrain complex and the mesopontine complex. The basal forebrain complex sends fibers to the cerebral hemisphere, and the mesopontine complex sends fibers to the thalamus and midbrain, including the substantia nigra and the ventral tegmental area, the locus coeruleus and the dorsal tract. So again, as you can see, these two main areas from which the cholinergic fibers arise and are distributed throughout the brain. But apart from these fibers, the long fibers, what we need to remember is most of the cholinergic neurons are interneurons within the area of the brain. And these neurons are involved in cognition, learning, memory, arousal, and motor control. Acetylcholine is one of the important neurotransmitters implicated in Alzheimer's disease. And believe that Alzheimer's occurs due to degeneration of cholinergic neurons in the nucleus basalis of memory. The substrate for synthesis of acetylcholine is choline. And the enzyme involved in the conversion of choline to acetylcholine is choline acetyltransferase. Acetylcholine is then degraded into choline by acetylcholine esterase. Now, donepezil and rivastigmine, which are both used in the treatment of Alzheimer's, are reversible inhibitors of acetylcholine. So, basically, they increase the availability of acetylcholine in the neurons and improve the cognitive performance. Coming to the cholinergic receptors, there are mainly two broad groups of cholinergic receptors, the muscarinic and the nicotinic. The muscarinic receptors are again of 5 subtypes, M1 to M5, and all muscarinic receptors are G protein coupled. M1, M3, and M4 are found in the cerebral cortex and hippocampus. And they play an important role or mediate learning and memory. M2 is present mainly in the basal forebrain. They function as autoreceptors. Muscarinic antagonists, that is, drugs which block the muscarinic receptors, include triacetylcholine, benzotropine, and they are often used to overcome the side effects caused by antipsychotic drugs. Tricyclic agents block the muscarinic receptors and hence produce some of their adverse effects. Since blockade of muscarinic receptors produces adverse effects like dryness of mouth, constipation. These tricyclics, which have muscarinic antagonistic properties, can produce these side effects. There is no muscarinic agonist in clinical use because the side effects probably would be intolerable. Coming to the nicotinic receptors, the nicotinic receptors are inotropic receptors, and each nicotine receptor has 5 subunits. Now, depending on the arrangement of these type and arrangement of these subunits, there are basically 3 main classes of nicotinic receptors: the skeletal muscle receptors, the standard neuronal receptors, and the homomeric receptors. The homomeric receptors have the same; all 5 subunits are the same. Now, the neuronal receptors of nicotine are widely distributed in the brain. They have pre, peri, and extrasynaptic, in addition to the postsynaptic. So, they can also function as heteroreceptors and autoreceptors. Nicotinic receptors have a lot of clinical significance in pathogenesis of psychiatric disorders as well as in treatment. The neuronal nicotinic receptors, they mediate some of the reinforcing and addicting properties of nicotine. And they also help in understanding the mechanisms of addiction. A mutation in the nicotine receptor subunit predisposes to schizophrenia. Studies have shown that the alpha Alpha-7 nicotinic receptors are potential therapeutic targets for the neurocognitive dysfunction in schizophrenia. There's a loss of high affinity nicotine receptor sites in Alzheimer's and Parkinson's disease. Varenicline, which is used in the treatment of nicotine use, harmful use of nicotine, is a partial agonist at the alpha 4 and the beta 2 receptors. Galantamine, which is used in the treatment of Alzheimer's, potentiates several subtypes of nicotine receptors and also a weak inhibitor of acetylcholine. Coming to histamine, the neuronal cells which produce histamine are restricted to the tuberomammillary complex of the hypothalamus. So most of the cell bodies are located in the hypothalamus, but the projections of these histaminergic neurons is widespread. Histamine is believed to regulate sleep and arousal, and also feeding and appetite. The two main roles of histamine in the brain. There are 4 receptor subtypes for histamine: H1 to H4. All of them are G protein coupled. H1 and H2 are postsynaptic, whereas H3 is presynaptic and serves as an autoreceptor and also a heteroreceptor. So they are present on the terminals of other neurotransmitters. H4 does not have much of an action on the CNS. It's mainly not present in the CNS. It's mainly involved in immune regulation. As I said, the clinical significance of histamine is mainly feeding and arousal. The sedative effect of some antipsychotic drugs like clozapine and some antidepressant like doxepin is mediated through the blockade of the H2 receptors. Pitolisant, which is a selective inverse agonist of H3 receptor, which means it has an opposite action at the H3 receptors, is effective in countering drowsiness produced by histamine and has been used in the treatment of narcolepsy and obstructive sleep apnea. Now coming next to the amino acid neurotransmitters. These are now being increasingly understood to play a very important role as neurotransmitters and also being implicated in an increasing number of psychiatric disorders. There are 3 main amino acid neurotransmitters: the glutamate, GABA, and glycine. Glutamate and GABA are the main ones. And as I said, they have been implicated in the pathophysiology of many psychiatric disorders, mainly schizophrenia, mood disorders, anxiety disorders, and Alzheimer's. Glutamate is the most common neurotransmitter in the brain, accounts for about 80% of the brain synapses. And as you can see, the pathways are widespread. All sensory efferent pathways, the thalamocortical projections that distribute the efferents to the cortex, the pyramidal neurons of the corticolimbic region, mainly associational and efferent fibers, the fibers innervating the cerebellar cortex and the corticospinal tract, all use glutamate. There are 2 main types of glutamate receptors: the inotropic glutamate receptors, of which there are 3 families: AMPA, malic acid or K, and NMDA. The unique thing about NMDA receptor is that it requires binding of glutamate, glycine, plus depolarization for its activity. So it requires these 3 events to occur simultaneously. The AMPA is primarily responsible for the excitatory neurotransmitter, whereas malic acid reduces the transmission, so it's an autoreceptor. The metabotropic glutamate receptors are G protein linked, and they are mainly 8 subtypes which have been identified. Now, the importance of glutamate lies in plasticity and its role in the two main things, plasticity and excitotoxicity. I'll cover these briefly. Uh, Plasticity plays an important role in learning and memory. Basically, it's the increase in postsynaptic or change in postsynaptic efficacy which occurs due to repeated use. In long-term potentiation, brief intense stimulation results in increase in synaptic transmission. And basically, this is mediated through the NMDA receptors. Ketamine and phencyclidine, which are NMDA receptor antagonists, produce a dissociative amnesia. Long-term stable low-frequency stimulation of the glutamated axons actually results in reduced efficacy, which is a condition called long-term depression. Changes in synaptic efficacy due to insertion or removal of AMPA receptors can occur, and this accounts for the learning or the unlearning which can occur in organism. The clinical significance of this lies in phobias, where extinction of conditioned fear by activation of NMDA receptors in the amygdala can happen. So, cycloserine, which is a partial agonist of the glycine modulatory site, facilitates extinction and has been used in addition to CBT or as an adjunct to CBT in the treatment of phobia. Now, coming to the next important role of glutamate, that is in excitotoxicity. Now, persistent stimulation of the inotropic glutamate receptors can cause cell death. A condition called excitotoxicity. Now, excitotoxicity has been implicated as a cause of neuronal degeneration in Alzheimer's. That's it. It's an important pathway for pathogenesis of Alzheimer's. Excitotoxicity has also been implicated in the pathogenesis of schizophrenia and other psychiatric disorders like major depression, autism. Now, memantine, which is a weak inhibitor of NMDA receptors, it reduces the sensitivity of NMDA receptors to excitotoxicity and accounts for its use in Alzheimer's. Coming to GABA, which is the major inhibitory neurotransmitter in the brain, it's synthesized from glutamic acid and it is broken down by GABA transaminase to succinyl semialdehyde, which is converted to succinic acid and it enters the Krebs cycle. Valproate is a competitive inhibitor of GABA T, so it increases the availability of GABA and hence its use as an anticonvulsant and also in the treatment of mood disorders. The GABA neurons are mainly widespread. They are mainly intrinsic neurons. In cortex, they inhibit the activity of the glutamatergic pyramidal cells. So they basically have an opposite effect. In striatum, they regulate the dopaminergic neuronal activity. There are two main types of GABA receptors, GABA A and GABA B. GABA A is an inotropic receptor. Its activation is caused due to hyperpolarization, causes hyperpolarization due to influx of chloride. Benzodiazepines enhance the response of GABA at GABA A receptor. As you can see, the benzodiazepine receptor binds to a different site at the GABA receptor than GABA, and it enhances the response to GABA. Barbiturates increase the affinity of the binding sites for GABA. Baclofen, which is used in the treatment of used as a muscle relaxant and also in the treatment of alcohol dependence, is a potent activator of GABA. Glycine is basically an inhibitory transmitter. Its importance lies in the fact that it's a aggregated chloride channel, and glycine is also required for activation of NMDA receptor. Strychnine is an antagonist of the glycine receptor. Now, coming to, as I said, the important neuropsychiatric implications of the amino acid transmitters. They have been implicated in the pathogenesis or pathophysiology of schizophrenia. I don't think we have time to go into the details. The slides will be available to you later, so you can see that they do play an important role in the pathophysiology of schizophrenia. Various studies have shown that. They also play an important role in the pathophysiology of anxiety and depression. Basically, GABA, which is an inhibitor, the dysfunction of GABA can lead to anxiety disorders. And there's a significant reduction in GABA and glutamate in the prefrontal cortex in major depressive disorders. And NMDA receptors like ketamine can improve depression. So all this indicates the role of GABA in the pathophysiology of these disorders. They also play an important role in the genesis of alcohol dependence. Now, coming to the next class, I think we are running short of time, so I'll just skip through the next few slides, give you a brief overview of neuropeptides. More than 100 have been identified. They exert both a direct and a neuromodulatory effect, and they act on multiple receptors, mainly the G protein receptors, and they regulate a variety of behavioral and physiological processes, including feeding, sex, sleep, learning, memory, and they've been implicated in many psychiatric disorders, including psychosis, mood disorders, autism, and dementia. Some of the important neuropeptides include the corticotropin-releasing factor, the thyrotropic-releasing hormone, cholecystine, neurotensin, neuropeptide Y, oxytocin, vasopressin. There are more than 100, but these are some of the important ones in the TNS. The TRH, or the thyrotropin-releasing hormone, has an important role to play in depression. TRF, it's long known that hyperactivity of the HPA axis has been implicated in depression, and TRF plays an important role in the pathophysiology of depression. Oxytocin and vasopressin, they arise mainly from the bed nucleus of the stria terminalis, and again they have widespread. Apart from, of course, most of the fibers arise from the hypothalamus, but there's another group from the bed nucleus of stria terminalis. The fibers which arise from the hypothalamus, the release of oxytocin and vasopressin into the pituitary mainly serves as hormone, but these other fibers are the ones which play their role as neurotransmitters. Oxytocin and vasopressin play a role in anxiety. Oxytocin has an anxiolytic effect, whereas vasopressin has an anxiogenic effect. Oxytocin also plays an important role in social cognition, concept of maternal behavior, social attachment, and attraction between mates, and it facilitates female sexual behavior. This regulation of oxytocin has been implicated in autism, depression, and anxiety. There are other neuropeptides like neurotensin, which modulates the mesolimbic dopamine receptors. The cholecystokinin receptors, which have a role in the pathogenesis of schizophrenia and panic disorders. Substance P has been implicated in major depression and PTSD. Neuropeptide Y has again been implicated in depression and PTSD. Neurotropins are polypeptide growth factors which influence the proliferation, differentiation, survival, and death of neuronal and non-neuronal cells. So basically, because of this role in regulating connections and neurotransmitter release and synaptic structure, they have a role to play in long-term potentiation and synaptic plasticity. These are some of the important neurotropins: the nerve growth factor, brain-derived neurotropic factor, neurotropin 3, and neurotropin 4. Their clinical significance lies that they have been implicated in the pathophysiology of a wide variety of neurodegenerative and psychiatric disorders, including Alzheimer's, Huntington's, Parkinson's, depression, anxiety, and substance abuse. We'll talk a little bit in detail about nitric oxide, because nitric oxide is a novel new neurotransmitter agent. There is no receptor for nitric oxide. It diffuses directly into the cells and acts through two pathways, the cyclic GMP and the S-nitrosylation pathway. Ultimately, it acts through the second and third messengers by activation of protein kinases, altering the protein function, the new gene expression, and the long-term potentiation and long-term depression of the neuron. Nitric oxide has an important role because of their role in all this. It plays an important role in learning and memory, neurogenesis, and in the pathophysiology of neurodegenerative disorders. Inhibition, nitric oxide is synthesized in the cell by nitric oxide synthase, which and inhibition of nitric oxide synthase enhances the effects of cocaine and amphetamine. Also, animals which are deficient in nitric oxide synthase display aggression, and hence nitric oxide may play a role in affective disorders. There have been changes in the nitric oxide synthase levels in the postmortem brain samples of schizophrenia. Now, I think carbon monoxide, there is not enough time, so we just skip through it. Just remember that it has an important role to play in olfactory neurotransmission. And in the roles of some of the roles of carbon monoxide, odorant perception is of course the most important, but it regulates a variety of perceptual and cognitive processes. It helps in adaptation to chronic pain, and it facilitates long-term potentiation in the hippocampus. It may have a role to play in anxiety and also in the sleep-wake cycle. A little bit about endocannabinoids, because these are important neurotransmitters. For a long time, it was thought that there was no cannabis receptor. For the cannabis receptors which were discovered, the endogenous substances which acted on them were not discovered till very late. Now, basically, we know there are two cannabinoid receptors, CB1 and CB2. CB1, which is a G protein coupled receptor, is predominantly present on the axons and nerve terminals, and it has an important role in regulating neurotransmitters. CB2, there are few receptors in the brain. Now, there are 5 endogenous cannabinoids which have been found, which act on these cannabinoid receptors. The first one was anandamide. Later on, 3 or 4 others have also been detected. Endocannabinoids are basically all synthesized from arachidonic acid. They are not stored but are synthesized on demand, and they are degraded by the enzyme fatty acid amide hydrolase. Endocannabinoids are basically inhibitory neurotransmitters. So that means that inhibition of GABA by endocannabinoids can actually lead to excitatory effects. That is what I told you in the beginning. Increase in release of brain endorphins and increased dopamine release in the nucleus accumbens is one of the effects of endocannabinoids. They have many roles and clinical significance. Endocannabinoids play a role in synaptic plasticity, both long-term potentiation and long-term depression, and hence they probably have a role to play in memory. They are important regulators of anxiety. Like FAEH, which I told you breaks down cannabinoids. So FAEH inhibitors reduce anxiety in animal models. Endocannabinoids also have a role to play in forgetting painful memories. So cannabinoids may have a role in the treatment of PTSD. Endocannabinoids also have a role in mood disorders. They have a role to play in addiction, and cannabis, it is known, can produce psychotic symptoms and precipitate schizophrenia. So these endocannabinoids may also have a role to play in psychotic disorders like schizophrenia. Cannabis is known to be an appetite stimulant, and rimonabant, which is a cannabinoid 1 receptor antagonist, CB1 receptor antagonist, promotes weight loss, and it has been used as a weight loss agent. THC has analgesic properties, and it is believed that endocannabinoids may also have analgesic properties and may mediate stress-induced analgesia. It is known that stress in military personnel induces analgesia. It makes them more tolerant to pain, and endocannabinoids may mediate this action. I don't think we have time to go into eicosanoids, which are also newer neurotransmitter agents. But basically, what you need to know is that they are concentrated in the neuronal membrane, and they increase serotonin levels, enhance dopamine transmission, and regulate corticotropic releasing factors. And they have a role to play in mood disorders. It's known that omega-3 fatty acids have a protective role in depression, and they've also been used in the treatment of depression and in the management of negative and positive symptoms of schizophrenia. That's about it. Thank you. I think we are running short of time, so that's why I had to run through the last few slides. Any questions you have, I can take them now.
Thanks a lot, sir. That was pretty comprehensive. In fact, there's 2 classes crammed into one, and sir has really done justice in giving one class itself. So, if there are any queries, please do ask. These are basic sciences and more of primarily of knowledge domain, not of experience.